Yes, I am live. Hello, everyone. Hi. Welcome to another Saturday microscopy live stream. Oliver here, Microbehunter here. Yeah, I'm getting started, and like every week, I need to get some housekeeping done first. So I always have to open a YouTube window at the same time to see if everything's working. I think it seems to be working. Okay, so yes. Okay, let's get started. I see that a lot of folks already started to join in the chat here. I'm just cleaning up my table a little bit as well. I've got so much stuff lying around here. Yeah. Hi. And today's live stream, today's microscopy live stream is going to be a little bit similar and a little bit different in the previous weeks because it's going to be a little bit like a variety show under quotation marks today. So I'll be talking about several things. I'll be showing you several samples a little bit. Usually in the last half an hour, I answer some of your questions, and this is going to be different today because my son, who always helped me to print out those questions, is not around today. So I will kind of be answering the questions, also in between. Yeah, so it's going to be kind of one continuous interactive live stream here. So like every week, I would like to say hello all around the world. There are already a couple of questions right at the beginning here, which I'm going to comment on. So hello and everyone from Vietnam. Okay, I should not change the trailer. Just in case you're kind of wondering, the trailer which is playing before the start of this live stream, that is from the YouTube audio library. This was a piano music piece. Yeah. Good morning, everyone. I have to tell you, good evening in Central Europe, where I am right now. It's kind of late in the evening already. Hello from the Belgian coast, from Hamburg in Germany, from the Philippines, from the UK, from Belgium, from Denmark, from the Netherlands, from Syria, from the United States, from Greece, from Finland, from the Philippines, from the United States, from Wales. Wales is in Austria. It's not so far away from where I am. Okay, everything is fine. Okay, audio is also good. From the United States, hello. From the UK, Gainesville is FL is Florida, I suppose. Yes, and from the United States again. Hello, everyone. Okay, it's really nice. I need to change this from top chat to live chat so that the sequence is correct. So what am I going to do today? I have to tell you the following. Look what I've done here, and I want to explain this a little bit. These are microscope slides which are made actually already several years ago. Let's make it a little bit bigger, right? Back in 2024, last year around. And I added a QR code. There is a reason why I've done it. So it's kind of a little bit like a suggestion that I have. And so maybe if there are some kind of big slide making companies out there, maybe you want to kind of pick up on that idea. So yeah, steal my idea. That's basically what I'm saying, to the benefit of the whole microscopy community. Because what I'm going to show you is this kind of derived a little bit out of a frustration that I felt. Okay, I just want to show this to you. So frustration number one is the following. It's not a big frustration really. I've got a whole bunch of slides here. Look, look at this. Yeah, these slides came along with a microscope that I bought. Okay, and no big problems here other than that it's basically labeled in Chinese. Yeah, so that is not the main issue really. There's a fairly easy, workaround here. I mean, many of the slides you just know what it is anyway because they're insect leg, for example. But I want to show you something, and that's the following. If you just take one of them here, and if you take your mobile phone, let me switch off the thing here, and if you go to camera, does this work? If you go to camera, and if you take a picture like this, then it will actually translate the whole thing. So B's third pair of thorax and legs. So it's basically the third pair of a leg of a bee. So this is kind of pretty nice, this Google Translate app. But that's actually not the point of those QR codes here. We can actually look at this here as well. This one, what is this one here? What does this say here? Yeah, these are testicles. Yeah. It doesn't say from which animal though. Next one, what about this one over here? skeletal muscle, longitudinal, I don't know if you're able to see this, skeletal muscle, longitudinal, crosswise, cross-section, yeah? So that's a skeletal muscle. I kind of like this, okay? And why am I showing this to you? Because, yeah, this is actually also an opportunity to get some other microscopy stuff translated, like, for example, in identification books and so on, yeah? This slide, what does this here say? Onion skin, yeah? I think the translation is pretty okay. Okay, that's not my complaint here. I've got another problem really. Let me move this further up here. And what does this say here? Dicot plant stem cross section. That's also fine. Okay, so that is actually quite a nice workaround here. But I'm going to show you now the following. And this is not so much only with this here, but essentially I bought myself a very nice slide set, which I want to show you. And then I'm going to also, I'm quickly putting those other slides here back here, because these other slides here, they are really nice as well. However, it's a little bit frustrating. And I want to show you why this is the case. So first of all, a very nice slide set. I just show you how it looks like. Yeah, it's from the company Konus. They're not only selling microscopes but also telescopes, and they have a slide set. And this is not just a regular one; it's a pathological one. So basically, it contains, apparently, human body tissue, which basically is diseased. So basically tumors and so on. So I know it's probably not a nice slide set. So it's used for medical training. And then it came along with a really nice booklet over here with a general description of the contents. Also no complaints about that. Yeah, so basically there are 10 slides here, yeah, in different languages. Yeah, and then there is also basically a catalog here of other slides. Yeah, so, but I'm going to show you the following now. I think in all sorts of different languages. So I like that idea. However, I do think it's a missed opportunity. And I would like to kind of show this to you a little bit. So I'm going to pull out one of these slides here. Look, it even came along with some empty slides. And even here, this one is quite nicely labeled. Okay. So these are basically, it's liver cancer. I know it's not a nice example here. It's described in all sorts of different languages. I'm going to show this now to you under the microscope, and then maybe you will get my point, because you're probably going to feel pretty much the same way that I felt. Okay, so let's here we go. Okay, I'm going to put this now under the microscope. So basically, that's a liver tumor. That's how it looks like. Oh, thank you very much for the donation. This is very nice. By the way, if you donate money, all of this money is used for microscopy. Okay, so I'm buying more slides and so on that I can show you. And this is basically, let me go into bright field. Okay, so that's basically a so-called secondary tumors of a liver. And I just want to show this to you. And then maybe you're able to follow a little bit the problem that I've experienced, and you're probably going to feel the same way. So that's basically how it looks like. Okay, yeah, okay, that's basically liver. Basic secondary tumors means that there has been in this person some kind of a tumor somewhere, so an uncontrolled growth, and then the cells broke off and they were carried along the bloodstream, and then they settled in the liver. And in the liver, these cells also started to continue to divide to form secondary tumors, and this is how it looks like. Okay, and okay. I mean, I know a little bit about these the structures that you see, the cells, the sinusoids here, but honestly, where's the tumor? Yeah, I cannot really make a lot of sense of that, because I'm not a pathologist and I'm not really trained in that. So I bought myself a slide set now, and I don't even know what to look for. Actually, today in the morning, I did some research. It took me maybe 15 minutes to, also with, honestly, sorry to say so, also with the help of ChatGPT, which kind of directed me to certain sources as well. I was able to determine, actually, and learn. I was able to learn that indeed, Let's turn on the arrows again. Those structures over here, these are the secondary tumors. Yeah. And because the nuclei are much bigger, they stain differently. We can zoom in even more, but you don't know that. You don't know that unless you've got some kind of specialized training. Yeah. So the cells here are much more irregular, for example, and so on. So this is, yeah, Marie is saying these slides should come with a healthy sample so one can learn to compare. I think I totally agree with you. And that's the whole point. Later on, I'm going to show you actually where you can see a difference. But this is a little bit the frustration that I, as I'm a biologist, right? But I'm not medically trained in histopathology, right? So that's a little bit the problem that I, in that sense, I'm a lay person. I cannot really make sense of what I see, right? Because I cannot compare. And there is no description. The description which is included is just a general description, but it's not slide specific. So I had to actually dig in and do some research myself to kind of, and then sometimes the pictures that you see online are not actually the identical than the way that we see it over here, right? But I was able to find similar pictures which actually illustrated that these structures here actually are basically metastasis, yeah? So where doctors know that this is basically, that's the problem. But honestly, I don't know that. Yeah, or these things here, right? Yeah, these the cells stain differently. Though that's basically that's a tumor tissue. But honestly, that is the thing here that yeah, I'm not able to know a lot about this, right? And that's the reason why I've had the idea of putting a QR code on some of the slides, and I want to show this to you. There is now a question. I wonder how these tissues are sampled. There are surely ethical considerations involved in this. This is yes, an interesting point, and indeed a couple of months ago I did receive emails from a concerned person who basically said, yeah, well, how does this actually work? I got myself a slide box with healthy samples, right, of human tissue. Was consent given, or how does this actually work? And I can only tell you the following. Well, honestly, especially in medical training, there are so many of these samples, right? And what they do is at hospitals, they take so-called biopsies. So they take a sample, for example, and then they process the whole thing and they put it under the microscope for diagnosis. Yeah? So this is actually something that is done normally. So the whole ethical debate is a valid one, of course. However, I personally, I would say I don't have such of a huge of an issue because, especially in the medical training, I mean, you're dissecting bodies. I mean, there's lots of these things going on, so this is something that is especially when there are organ donors who are consenting to this, I see less of an issue. But I get the idea that some people might consider this a little bit strange to actually know that, well, I'm looking now at human tissue of a person that might have died from this disease, right? But my point is that's a different point, right? But my point now is the following. I want to learn something from this here, and I feel a little bit limited in my learning because I cannot make sense of the things that I actually see here. Yeah, seems histopathology is not for amateurs. I probably would agree with you. I mean, that's why you go into medical training. Yeah. So this is indeed a valid point. But you see, even I, as an amateur, I want to learn a little bit, yeah, about how these things look like, right? So this is one of the things I want to show you now, where I do have a comparison. And I know this is also a little bit. Some people consider this weird, but actually, it's one of the nicer specimens to look at, because what I have over here is this slide. I want to show this to you now, because here I can actually explain it to you, and the reason why I can explain this slide to you is because that's part of the syllabus that I'm teaching as well. But I have to find now the correct one over here. Look, which one is it? So that's some kind of a tuberculosis. That's not the one. What is this here? That's a lung, basically, where as carbon has been inhaled. We might want to look at this as well. You can actually see the carbon particles. Okay. So this malaria is on here. I had a problem finding the malaria parasites. I'm not that trained yet. That's basically a lung. Ah, that's the one here. That's cancer of the testicles. Yeah, so what I would like to show you now is I want to show you intact testes and a tumor. And here there is actually a difference visible, right? But it's only because I know very basic stuff here, because this is part of the biology syllabus. I'm teaching, for those of you who don't know, I'm teaching an advanced biology course as well in school. So I'm going to go to the scope view again. I'm just going to explain. I made some videos, by the way, for that very reason, because the process of spermatogenesis, the formation of sperm cells, is actually something that students need to know. Yeah. So I'm just going to show you how intact testes look like. Let me just find a how do you say, a good place here. These are not so. Yeah, this one over here, right? Yeah, let's use this round one over here. What you look at, what you're looking at here are so-called seminiferous tubules, and then I have not forgotten the QR code. Okay, so this is all kind of leading up to this whole QR code idea. So what you see over here, these are cross sections of intact testes, and what you have inside here, these are the sperm cells, the spermatozoa, and they're formed here in the wall of the testes. So these round, and here those little fine lines that you see here, these are the tails of the sperm cells. Right. The idea is let me go out with the magnification. The idea is a little bit so that you see a little bit the overall structure of how intact testes look like. And in the cells between those seminiferous tubules, this is where the hormone testosterone is formed. Right. So that's just the reason why I'm showing this to you is because, for comparison. And I'm going to show you now over here this slide with testicular cancer. Yeah. And if you look at that, it looks like this. Yeah. It looks very different, right? Because the tumor cells are growing all over the place. Yeah, I mean, and if you have something like this, then you can actually compare it, right? And I think everyone can learn something. Well, if you've got a tumor somewhere, then the cells are growing in a very irregular way, and the normal structure of the organ isn't there anymore, right? So I think we all agree that this one here looks very different, yeah, than this one over here, yeah. And I think we all have learned a little bit that way, right? Yeah. So that's essentially the thing. And now I'm saying is, yeah, sure. Now, because I know a little bit, I can show you some kind of comparison. So, and what I would like to therefore propose is the following. Okay. And I hope that maybe some slide makers or slide manufacturers or some companies, please steal the idea. Okay. I don't have a copyright on the idea, right? I'm just going to show you now the following. Let's have a desk view again. Why not the following? Okay, why not put a QR code on a slide? Okay, and when you do the following, then you go over here to the picture. Okay, let's have a look. When you go over here to the, it's not in focus. Okay, when you basically see, it's kind of also reflecting the light. Okay. Here we go. And then it opens a link. And then it opens a homepage, a website. In this case, I just linked it to one of my pages. And then you get a picture of the slide. And not only that, but also a description of what you see. And maybe, here on the bottom, also some slide information. I mean, this would be cool. Maybe also some comparison pictures that you could include here. Yeah, basically what you see. Of course, every slide is different. I get it. But I mean, you can also add some labels here if you want to. Yeah. And then you have some basically some description of the slide and maybe some additional information on the slide that you're not able to fit. Yeah. On the label here, right? So this is something that I kind of propose, which would kind of, I think, would really greatly increase the value, value of a slide. Yeah, just saying. Yeah, what are the black dots? Which black dots? I don't. Yeah. So those dots over here, these are the individual mites. I can also show them to you, right? So if you want to. So, or for example, over here, I made just 3 samples here. By the way, if you visit my webpage, I just need to say this. Look what it says. if you visit my webpage, the microbehunter.com, it's giving me this error message, and I think I found out why. And the reason is there are, I don't know, I think I've got some kind of somebody's trying to take down my webpage. There are too many requests, yeah. So that's a little bit, so you got to try it again, and then it will, until it works. But I want to show you over here. Let's go the next one here. Yeah. And it opens up the next link here. Yeah, I have to reload it a couple of times, hoping that it will work. Okay, and this is the stinger of a wasp. Again, yeah, that's by the way, I prepared this a couple of years ago, right? It's a stinger of a wasp, and then there is some descriptive text about that. Yeah, the server down. Yeah, somebody's for whatever reason, yeah. Who will pay for the servers? Yeah, well, that's another question. I mean, if there's a company that is doing that, then they should basically, they'll have their own servers anyway as hosting providers, right? Because people are hosting websites anyway. Yeah? And that's not really expensive because web pages have to be on a server in any case. And in this case, I put it simply on my hosting provider. Yeah? But where is let's go into the picture again. Yeah. This one over here, last example that I just want to share with you. Yeah, this one here is a tiny little mite. It's not a tick, yeah, but it's a mite that I found some time ago, and you can also give some information. Yeah, QR code to Wikipedia entry. Yes, also a possibility. At least then the Wikipedia entry has to be edited in such a way that it's actually relevant, of course, for the object. But there are many possibilities here. But I think if you simply, I think that would be a nice possibility how you could actually, basically extend the value of those slides simply by providing more information to that. I hope that somebody's going to pick up on that idea, because otherwise we're all going to be confronted with really beautiful slides which essentially don't tell us a lot because we don't have the training. So did you make the website myself? Yes, it's basically the website microbehunter.com is hosted, and I bought a template and I basically fill it up. So the whole thing is mostly mine. So basically I'm managing the website as well. So, and the reason is there's too much website traffic. I don't know why it comes and goes, but it's a different issue. The companies probably don't do it out of the fear that the slides won't sell anymore. You look at the pictures on the site. The thing is the following: what one could do, however, is if you make the website with the slide information, if you basically put it a secret, how do you say, a secret combination? I call it now varroa mite, or just mite, or wasp stinger, or whatever, right? But if you give it some kind of a combination of characters and numbers that, just like for example for YouTube video, which is then essentially nobody knows about that, and then only those people who actually have the slide can access that information, because so that's simply an idea that I have. I just want to float it, because I think that would be kind of cool. I'm just saying, but I don't have, unfortunately, I'm not selling slides or anything like that, so I don't have the capacity to do. Will there be some copyright issues? The thing is the following: if you cannot copyright nature. So the thing is if I unless I know that some companies don't like it if slide companies, if you scan the slide and then make them available, right? But if those very same companies then essentially enhance their slides by adding additional information, and I don't, it's of course it's not a copyright issue, right? Because it's the companies. If I basically I made those slides, I collected those samples, I made those slides. If I basically now were to sell slides with samples and kind of include the information that kind of links to my homepage or so, there are definitely no copyright issues, because you're asking, so this is a little bit a thing that I just wanted, an idea that I kind of want to float here. I don't know. Let me hear what you want to say. So that's number one. I also have a couple of other things I want to show you today. So number two, the info linked to it. Oh, okay. Got it. I get it. Yes, this information. Okay, so this is basically, actually, this is the picture of the mite here that I've got here. If you want, I can actually, it's tiny. You don't even see it, somewhere over there. Let's have a look. Mites are tiny arachnids related to spiders and ticks that live in many different environments, including soil, water, plants, and even on animals. Most mites are only visible under a microscope. They have 8 legs and a small round body. This is a very general information. I mean, what you could do, of course, is you could maybe add some labels here, right? Maybe even give some for students, maybe some instructions, try to count the legs, whatever, right? So what about this information here? I tell you why there is no copyright problem. Because, you know what I've done? Who wrote this article? ChatGPT. I actually, into ChatGPT, I told ChatGPT, please write me a summary of mites, including the following information. So you give it an outline, and then ChatGPT will actually convert it into a readable text. Yeah. So you don't copy paste this from anywhere, right? So in that sense, there is no copyright problem. Yeah. So this is just something that I actually wanna show you. Yeah, kind of as a little suggestion here. I'm reading again the thing here. I recently watched. Okay. Okay. There's some other comments going on. Okay, so that's basically part number 1. Okay, that I just wanted to share with you. So number 2, and I'm not quite sure whether I should do that or not, and I just am again floating that idea, and then we can see how it's going, where it's going to take us. Okay, the thing is the following. Some of you might know or might not know that several years ago, maybe about 7 years ago, 8 years ago, I for a few years, every once a month, I published a magazine, a freely downloadable, a PDF magazine. You can still download it from the website, from the microbehunter.com website. And basically, and it contained articles written by the community, right? So people sent me pictures and articles, and I simply did a layout and made it available for download, right? So that's basically what I've been doing for a couple of years. And then this was actually quite a lot of work because I had to read everything and edit everything more or less, and then do the layout, and then I basically put it online for download, and more or less a whole bunch of people downloaded it. And then basically it was so much work that I changed over and made the YouTube channel that you're basically in right now. But I was kind of thinking, if there is the interest to release a magazine free, of course, of charge, with contributions from the community, maybe once a year or twice a year, then that is manageable. Okay, I'd be collecting articles and I'd put it into a nice layout, and then people can download it. If there is a wish to do that, or a desire to do that, or an interest to do that and to contribute articles, then we can have a discussion about that. Right. So if you say no, it's actually videos are fine, and we've got a forum, and a forum is much faster anyway, and some kind of a formatted PDF, which you can also, if you want to, order in a printed form if this is not needed, or considered important, not right. We don't have to do that. But I'm just floating also that idea a little bit, that if there are a couple of people in the community that would like to contribute articles again, maybe we can restart this magazine concept. Yeah. Again, basically, I just collect articles and some pictures, and I put them into a nice layout, and then it can be downloaded. So I'm just also floating that idea in case there's an interest. Yeah. So there are again a couple of. Let me see if there are some more questions. Or hello, QR, great idea. Okay. Hi Oliver, what's up? I'm fine, thank you. Okay, so this is just something. So let's go on a little bit. What else can I do? I know I just recently made a video, but I just about that. So why not? Okay. For decades there has been a German magazine called Mikrokosmos. I know it. I've read a few of them. Yeah. it is somewhere freely available to download. Really, they make it free. The Mikrokosmos has been the German. It's a small A5 sized magazine, fairly thick, and it was a little bit on the level between amateur microscopy and hobby microscopy to the more science related aspect. So it was kind of a little bit nicely in between. So it was a very nice magazine, but they stopped publishing it a couple of years ago. Yeah. May I ask to introduce the software for producing the QR code for microscopic slides? So producing a QR code is quite easy. There are many free online tools available that do that. You just copy paste the web address and then it will create a QR code. Yeah, it's also possible in Chrome, the web browser, to directly create a QR code from the website that you're visiting, right, of the URL. So there are lots of freely available options out there. Yeah, you just have to Google for QR code generator, and then you can change the colors and the size and pretty much a lot, right? So this is yeah, I just used a freely available version online. Yeah? So I know that I've been doing a lot of talking now. Yeah, just saying it's a little bit of a variety thing that I'm doing today, and I said this because it's kind of fun. Again, look, and again, I just recently made a video. Does this look familiar? How many of you have already seen the video? Let's have a look at it. Again, I found a piece of wood. Okay, and look, let's put it under the microscope just for the fun of it. Okay, and yeah, let's get started with that. Okay, I just want to show you, because actually people, and I myself included, like to do a little bit of casual exploration, and yeah, why not? Yeah? Unless you have any other suggestions. So you know what else I have here, in case you wonder, where is it? Where did I put it? Where did I put them? Yeah, look, here it is. Do you know what this is here? This is basically for my coffee machine. I like to drink coffee. Look in here. I love it. It's the inside view. It looks green. There's some stuff growing on the inside here. I also want to have a look at this. Okay, so we're gonna put some stuff from the coffee machine, and something else I've got here. You know what this is? You don't know. This is how do you say, meat, artificial meat, which is made of soy products. And if you put that under the microscope, sometimes you also find some interesting things in there. Yeah. So, but for those of you who didn't see the video, and because I also am a little bit interested in finding out, look, you add a little bit of water. Maybe that's already enough. Yeah? I'm going to use this here as my, which one you already know that from last week, this homemade stand. And then, and now I dropped it, of course. Why not just take it in your hand like this, and you basically do the following. You just take a little bit of stuff, and you just rinse off whatever might be on here. Sometimes there's interesting stuff on here, sometimes not. Okay, look, there is actually quite a lot of stuff in here as well. In the meantime, you know what I might do? I'm going to take maybe some of the solid material from the way of the bottom. Yeah. And then let's try our luck here. I put this in again. You might want to wipe here. And this is basically, a demonstration to show that you don't always need pond water. But when you add water to a piece of wood, just make sure that most of the wood is actually not underwater, but exposed to the air. We drop a cover glass. Yeah. I was surprised how little water you put in the tube in the video. Yes, and the reason why I only put very little water in here is because the water is going to be soaked up anyway. I want the wood to be moist, but not be completely submerged in water, because I want enough oxygen to reach the wood, right? Because microorganisms or whatever I find on here, they need oxygen, right? And if you flood everything with water, I mean, if you want to, you can dump it into water the very first time, but then later on you just let it soak for a day or two, or 3 days. So So let's put this away again. And we're just going to go into the scope view, microscope view. I don't like the arrow. Yeah, and let's have a look. And occasionally again, tardigrades is usually something that you might be able to find. Yeah. Yeah. So this is basically, that's now I'm using my 10 times magnification. That's how it looks like, right? So you see plenty of life. Yeah. And usually what I do in this case is I just scan through the sample like this, and I try to find something which is hopefully interesting. Why am I showing this to you? To illustrate also a little bit, you don't always need a pond nearby, to find microorganisms. And then if you're lucky, you're able to find also occasional tardigrade, a water bear, sometimes rotifers you find. Yeah, and that's basically what I sometimes like doing. You just put stuff under the microscope like this and just have a look at what I'm able to find. Yeah. Yeah. So there's a comment here, a nice comment. You make some really interesting videos. They're really good. Thank you very much. But, yes, I'm very happy that you like the videos, but the views have been going down. Yeah, the videos are not quite as I don't want to complain, right? The community is very interested and engaged, but honestly, if I'm lucky, I'm getting 1,500 views per video. And this is not a lot, and I've received the feedback already that some people are, and I totally understand it, some people are getting sometimes a little bit bored by the videos because it's kind of maybe interesting the first time, but then you put water samples again, it's kind of always a little bit repetitive. So I get it, and I feel so as well, and for this reason I've been thinking, but I'm not quite sure of whether it's a good idea or not, to broaden the videos with topics that are maybe microorganism related, but not specifically microscopy related. And I don't know if this is a good idea or not. So if I talk about general microbiology stuff, which is slightly more theoretical, okay, I don't know if this is a good idea or not. If people are, because sometimes I don't have microscopic footage to show you. Okay, I give you an example of a topic. Maybe is how can it be that there are more bacteria growing on our body than we have body cells, right? Yeah, and then I can explain a little bit how scientists have determined that, right? But you see, it's not so much microscopy related; it's more microbiology related, or maybe why are cells so small? It has to do something with the surface area to volume ratio. It's a little bit more biology, that would actually broaden the different topics, okay? But I don't know if it's a good idea or not to go into that direction. I really don't know, and I would be very happy for your feedback. Okay, so let me quickly go up with the magnification a little bit, and then let me have a look. And as we enjoy the movement of those little microorganisms here, okay, let me have a look here. Okay, what you've been reading, writing here. Okay, there are lots of points here. Microcosmos, yeah. May I ask to introduce the software? So these are basically I used I think 2 or 3 different free online websites for generating QR code. Free QR code maker or generator. You just Google for that, and then you'll find many examples. Okay. We'll watch it later, no matter. Okay. You can also make them through the programming language with literally 2 lines of code. Oh, okay. This makes you independent of any charges QR code website might give you. Yes, it is also possible that, for example, in Chrome, you can just visit a web page and then directly make a QR code out of it. There is actually in the menu, it says show QR code. It does not give you a lot of options to format it though, to change the appearance. Which data is coded in the QR labels? Well, you can code whatever you want to code, text, numbers, but in this case, it's simply the website, the address of the web page that gives you the information and the picture. So it is not the actual information that's on the QR label, on the QR code, but just the link to the website. So you can link it to whatever you want to link it. Yeah. So yeah. You have a lot of flexibility. Yeah. Hey guys, with a revolver with 4 lenses, what magnifications do you have installed? For example, 4 times, 10 times, 20, 100 times oil, or 4, 20, 40, 60, or okay, yeah. Which set is best for you? I'll leave this up to the community. I have many of you already know my opinion. I can, if you want to know, which I think 20 times is very suitable for observing water life, because just from the size, it gives you enough magnification to see more of the cells in detail, but not too much, right? So that they don't move out. Yeah. I generally do not use oil very often. Oil, 100 times oil. I do not need that high magnification. I use 60 times a lot, also because it's easier to use, right? I don't have to use oil immersion. So that's, yeah. I would just say 4 times definitely 4. It's a question of taste, I would say. Yeah, 4, 20, 40, 60 is fine. 4, 20, 40, 60, it doesn't really matter a lot, I would say. Yeah. Theoretically, if you have practice, you can even not have a 4 times objective. Yeah. Okay. So I'm continuing to read here. I have an old water sample that is full of bacteria. What should I do with it? So basically, you want to think about disposing it. You'll be shocked. You dump it down the toilet. You rinse it out with hot water and you kind of clean the jar. That's it. Yeah. There are lots of bacteria. Honestly, if you're worried about killing the bacteria, let me tell you, in the sewage water, this is where bacteria live, right? You're basically, just. Yeah, it's not going to make a huge difference. So just dump it down the toilet. Yeah, I would say. Yeah. Okay. So that's. I'm just reading here. Okay. Yeah. Which magnifications? Have you had your coffee? Well, yes. Already in the evening, I also have a cup of coffee. Yeah. I would appreciate knowing more about microphotography techniques. Okay. Thank you for the recommendation. I can actually show you how I do actually I have to say photomicrography. That's the way I'm supposed to say it. Photomicrography. Okay. How is the wood sample any different from the pond? Well, let's put it this way. Every sample is different. And what I do sometimes find on the wood sample, because the lichens on it, I do find occasionally some tardigrades, right? So not here so much. Yeah, but look, I mean, this is really massive. Yeah, there is really lots of stuff going on here. Yeah? And certainly, if you were to do some kind of an analysis, taxonomic analysis of the cells that you see here, obviously there are probably going to be different species here. Yeah. Okay. I prefer if you do stuff that you are interested in and have passion for, not making videos for clicks only. Yes, thank you for this feedback. I feel the same way. The thing is the following. I also would like to because I'm interested in so many things, it would be nice if the community also is interested in. I do not want to do stuff that kind of bores the community either. But it is correct as well that because for me it's also a hobby, I really do a lot of stuff that I'm primarily interested in. So I basically agree with you, but it would be nice if basically I would, to a certain extent at least, or in some cases, also meet the how do you say, the interest, of the community. Yeah. So for example, because you asked what can I find on the wood here, I made a video about this, right? And some of you have already seen this. For example, those bright or round structures that you see, these are spores of fungi, right? And those fungi, of course, you find a lot on the wood, but not so much, of course, in a pond. It seems that the full image of specimen embedded in a microscopic slide and could be seen by scanning of a QR label by a mobile set. Is it the same? The full image, you will not be able to encode the full image on a QR label alone. This basically because that would be like having the full image, yeah, in small. Then you might as well just put a picture on it. The advantage of a QR is that you are able to get the text, or in this case, the link to the website, even if the QR code is a little bit damaged. Yeah, so there is a little bit of redundancy built in. So even if the QR label is not perfect, and then sometimes it's still able to be read. So that's the whole point. And actually, I think it's much more convenient if you just have a link, because then you can have much more. How do you say? You can pack much more information on the web page, right? Links to other websites, YouTube videos, whatever. Yeah. So this is quite a lot of stuff going on here. So you see, this wood is super active, and the thing that even surprises me a little bit is I mean, look, this wood was completely dry. Yeah, and then I added a little bit of water, and a few days later, this is how it looks like. Yeah. So this is the thing. So somehow those microbes must have survived somehow. Yeah. So DYS, I think you may mean to say do-it-yourself microscopy props. Okay, yes, some upgrades maybe. I've got a 3D printer, so it is possible to do a little bit, and I'm going to show you something else that I've done, because I do want to comment on the very, very first question in this chat, which is referring to a video from Steve Mould, which is who's a famous YouTuber, who basically also presented a new microscope. And when I saw the video, I said, I want to have that microscope. But it's expensive. Do you use methyl cellulose to stop motion of protozoans? Oh, thank you very much again for a contribution for a donation. Yes. How do you prepare a medium to reduce motion? Okay. There's a question here. Do you use methyl cellulose? Yes, I use methyl cellulose, and yes, I have made a video of this. So, and this is a tube with methyl cellulose. So just a short time ago, if you actually check this channel, I made a video on slowing down microorganisms with methyl cellulose. Okay, so easy to make, highly recommended. You know what? Because you're asking, let's try it out, right? If I already got it here. Right, so methyl cellulose is a highly viscous solution, and it really slows down. Yeah. Eit Münster says hello to my favorite microscopy guy. I think I know who you are. Okay. Yeah. Thank you for joining from Eit Münster. Okay. Okay. So at Oliver, there are a mass of small dots visible. Is there something wrong with the cover slips? In my observations, I do not have this phenomenon. I noticed this last week as well. There are a mass of small dots visible on the cover. I do not have this phenomenon. Is there? There are a mass of small dots visible. Which dots do you mean? Okay, that's the question. It could be, I don't know what dots you're referring to. I don't think that you're referring to those moving bacteria right now. What you do is the following. Many of the cover slips, indeed, when they come from the factory, are not entirely clean. That's and you should actually dry wipe them. And you know what I'm going to do now is I'm just going to, for those of you who don't know the methyl cellulose, so there's several things I want to do now. The methyl cellulose one, the cleaning of the cover slips. And let's have a look. Just a second. I'm going to go back to the stage. Not stage, to the desk. Here it is. Okay, here we go. Very often, and this is a pity. Okay, if you actually, these are cover slips, and if you actually shine them in a light like this seems to be okay. But sometimes there is this, how do you say, white layer of stuff on here. And this is from the manufacturing, and it's many cover slips are kind of look a little bit, how do you call this? Like there's some kind of a grime on it, you know, and this is indeed something that actually disturbs the image quality. And so what I recommend is that you dry wipe it, or use some. This is dangerous, obviously, because you risk breaking the cover slip. The more expensive ones from brands and so on, of course, are completely clean, right? might not be necessary always if you dry wipe them, but indeed this is an issue, and I always have to clean the cover slips and microscope slides from the cheaper companies at least before using, because indeed they disturb so much that it's actually a pity, right? So that is number one. And let's try to slow down the microbes now using methyl cellulose. So, and what you do is, these are basically branded, the company sent them to me, so I'm just gonna use them because as a, and these were actually really nice slides because they were completely clean. And I'm going to now do the following. I'm going to show you how the methyl cellulose thing works now, right? Let's do this again here. Let's go in here a little bit to pull out a couple of more. Okay, see those thick things? So I think there should be enough in here now. And then what I do is, you always want to use a clean pipette for this. You try to, I misplaced the methyl cellulose tube. I cannot believe it. I put it somewhere on my table and I forgot where I put it. Just a second. This cannot be this can't be it. I've got so many things on here on my table that I actually forgot where I put it. Ah, here it is. It's in the test tube stand, test tube rack. So this is the methyl cellulose one. And what I usually do is I add approximately the same amount of this methyl cellulose. It's highly viscous. Okay. Maybe not enough, I don't know. Yeah, and you just add it. You gotta be careful a little bit because it does produce a lot of bubbles. Yeah, and you have to mix it properly. Otherwise, if it's not mixed, then yeah. I think maybe this is gonna work better. Yeah, but again, because somebody asked me to watch the video that I made a couple of days ago. It's deliberately an instructional video to show you how I've actually made this methyl cellulose solution, right? And then I'm just going to drop the cover glass on top. Okay, and then you would just basically put it under the microscope again. And then, if properly mixed, that's always a thing, if properly mixed, the microbes should be significantly slower. Scope view. So I have to find them. There are not a lot. Here's one. Yeah. And we go up with the magnification. Here it is. Yeah. And now you can actually also see the cilia, which are the tiny little hair, beating much better because it's, yeah. Let's go up here to change this. Let's go up with it's a little bit out of focus. Yeah. And remember, just before it was kind of swimming around rapidly. Yeah. Yeah. And the nice thing about methyl cellulose is it does not change the osmotic potential. So this means the cells, they don't gain or lose any water. I've tried it also with glycerin, which is also highly viscous. But the problem is glycerin, then the cells are going to dehydrate. They're going to lose so much water that they're going to shrink and shrivel, and then they're dead. Yeah. But methyl cellulose does not change. Change it. I'd have to, you know what, I have to really go down with the magnification now, because otherwise I'm not able to see. Maybe I did not catch a lot of these. That's again one of those demonstration effects. It's a bad sample I got, because I did not take any sample from, I should have taken a sample from the wood, which I did not do now so much. But I just took a sample from the water. And there were very few microbes. This is super disappointing, but this kind of illustrates a little bit of the demonstration effect, and I'm going to try it again. Okay? Yeah. Also, it shows a little bit that in different places you are able to find different things here. Let me... cover glasses can have stubborn stains. Try acid, vinegar, for example, because the vinegar will also remove some of the so-called the calcium carbonate, which are the so-called the water stains. Yeah. So yeah, let's add a little bit of water first here, and let's. Oh, you don't see that because I've got to go to the desk view. Okay, so add some water first, and you remember what I've done is I've taken some samples here and I've done this. Okay. Yeah. Maybe add a little bit more water. I need a little bit more liquid. Let's take some stuff here. This should do it. This should do it. So let's put it back again. I'm not going to put on the cover glass just yet. I just want to check first whether there is actually something there. Whether there's actually something there first. Yeah, there is slightly more life going on here. I think you see that, right? Yep, there is. Okay. Those big dark clusters are a little bit, it looks like a rotifer over there. Nematode worms. Yeah, here, this is a nematode worm, for example. Okay, so there two more nematode worms not moving. So you see, yes, this is actually a better sample because there is more movement going on here. Okay, so this means that I'll be adding the methylcellulose now here. Yeah, and then maybe, you know what, but for comparison, let's zoom in a bit more. This is, you're able to see now how fast they're moving now. Yeah. Okay. This here seems to be a dead rot for what we've got here. Yeah. So this is actually a better sample. Desk view. And let's do again the methylcellulose one. You have to take some time for it to be sucked up. Need some time to flow out. And carefully mix it to prevent, as far as possible, to prevent bubble formation. This is actually a very big, how do you say, spot? Very big. Yeah, so I think maybe enough. So I'm not going to clean the cover glass now because I'm too lazy. I'm going to just drop it on here. And we're going to give it a try. Okay, scope. Yeah, see, that's the thing is sometimes it's not much more difficult to see because so many of them are not moving anymore. Or not as quickly anymore. Here, that's a dead nematode. That's kind of cool. Now, the movement apparently is so slow. The streaming motion that you're able to see now is... Let's just have a look. Let's just zoom in into this one over here. Yeah, you actually, this one here, the round thing is still living, right? You actually can see the cilia beating, but it's not able to move at all anymore. Yeah. Yeah, so this, it's not dead, but now it's a little too, how do you say, too viscous. And now the cells are not able to move at all. Yeah, also the bacteria in the background. So maybe I did use a little too much of yeah. But you remember how it just looked like. I mean, was a heavy movement going on, very active, and now very little because this one over here, for example. Oh, where is it? Yeah, it's flowing because it's spreading out the, yeah. For example, this one over here as well. This is, again, the worm. You see, it's moving. The cilia are beating, but the cell itself is not able to move anymore. Yeah, yeah. So that's just just as a little comparison. Maybe I should have used a little less of the methylcellulose now. Okay. I use photo flow from the photographic film days for keeping water stains from the film. Works well on slides after washing. Place on a slide drying stand. Ah, okay. I remember this is because I also used to do some film development. So when I've developed film and then you added this kind of a drop of this detergent, which kind of prevented. Try it out because sometimes these detergents they will also harm bacteria and microbes a little bit because sometimes they will actually break down the cell membrane. Okay, because if they're a little bit acting a little bit like soap, you have to be careful, or at least you have to check that this whatever is left over on the slide does not harm. The why am I saying this? Because I remember during the days at the university when I was working in the lab, we used a dishwasher to wash our laboratory equipment, but we were not allowed to add any washing solution to it because any even small amounts that were left on the glassware prevented the microorganisms from growing. Yeah, so this was a very small amount, but this sometimes was even enough. Yeah. How do you change the cone of light reaching the stage from 4 times to 10 times by changing the condenser diaphragm? So the condensers, if your microscope has one, has a diaphragm lever, and by moving this lever left and right, you're changing the cone of light, and this allows you to control the so-called the numerical aperture and the resolution and the contrast. Yeah? So there is actually on the lever, and that allows you to change the cone of light. Yeah. It's interesting how much energy these microbes have. The smaller you get, the more dense water seems to be. Yes, water could feel like syrup for them. Yes, that is correct. This has to do something with the so-called it's fluid dynamics. That indeed for microorganisms, the air even feels almost like honey feels to us. Right, so it's yeah. I think it has to do something with this called the Reynolds number. Yeah, airplanes also are affected by this because larger airplanes behave differently than smaller airplanes, for example. Yeah, so this is also because size changes around the way that a fluid or liquid behaves. See, there is now no movement going on at all here. Yeah. Did you ever 3D print a little drying rack for slides and covers? No, I didn't. But actually, that would be a nice idea. Some kind of a drying rack. What I'm doing right now with the slides is I just basically put them on tissue paper and air dry them like that. Yeah. Why doesn't the methyl cellulose dehydrate the samples like glycerol does? Does that have to do with the added hydrophobicity from the methyl groups and sugar backbone? Good question. Glycerin in any case really is very hygroscopic, which means it really attracts water a lot. And yeah, I think I don't methyl cellulose is a polysaccharide. Yeah, and I guess maybe it also has to do something a little bit with the size of the molecules because these seem to be kind of long fibers, and then they're not able to impact so much on the osmotic potential, I guess. Yeah. Okay. Have you ever just put an uncleaned cover slip on the slide to see the dots? We can do that. Yes, I have done this as well. So I just want to show you. The problem is I've got all of my slides here are kind of clean already. Okay. But let's just try one here from the very bottom. Yeah. I think I've seen some. I need. You know what? If you just let me see if I just maybe wait a second. Maybe I'm able to find something here. I found something here. I've got some microscope slides here. I don't want to show the brand or anything, but just to show you that this is actually we can put it directly. Yeah, it's a fresh fresh pack of slides. Okay, it's actually one of the cheaper ones. Actually, these look kind of okay. Nah, actually not. Okay, so let's let's put this directly under the microscope. So this is directly from the box. Okay, it actually says written on here, cleaned and ready to be used. Okay, let's see. You decide whether this slide is clean enough, and we can already see from the reflections of the light that no, probably not. Okay, so microscope view again. So I'm going to put it directly on top here. We have to probably go up with the magnification a little bit. Okay. And let's go up yet further. Yeah, so this is basically how the microscope slide looks like. So you see there is lots of stuff on here. Yeah. You're probably not going to see it if you add other specimens to it, but actually I don't think that this is can be considered clean. Yeah. So that's a little bit, yeah, the problem. Yeah. So no, I think this is not a clean slide. And what I'm going to do now is I'm just going, and it's directly out of the box, right? I just open a completely fresh slide box. Yeah. So let's do the following, and I'm just going to dry wipe it. Dark field. Okay, I was wondering if dark field could. Let's try it. I only have a low power dark field. Okay, unfortunately. So with the 4 times, and I put in the phase contrast. I open this, and this is how it looks like. You know, you bet that dark field shows the dirt. Yeah. So this is not cool. Yeah. So if you don't want that, indeed, you got. I'm dry wiping it now. Desk view. I'm dry wiping it now. And then let's have another look. So it could be scratches too, not just dust. I could be, but it can be removed. Okay, experience has shown that it can be indeed removed. Of course, it's possible that these are scratches, maybe from yeah. But if you actually look at it now, then you actually see that it does look a little bit cleaner. So at least on one side. Let's have another look. Actually, I never tested it like that before myself, so it's quite. So this is the side that I have not cleaned, not dry wiped, and I'm moving now over. You can also see on the top here. This is now the side that is dry wiped, and it does look a little bit better. Yeah. Let me get this in focus. I think it's either the top or the bottom surface of the, yeah, either the top or the bottom surface of the slide. And if I move over here, yeah, you see that's the non-wiped side, and this one here is the wiped side. So it can be removed. Huh? I mean, I just have to say it now. I know it's. I'm not trying to make advertisement for the company right now, but I mean, I'm just saying that the company Globe Scientific did send me, and I didn't ask for they just sent it to me, sent me a pack of microscope slides, and they're completely clean. Yeah, just saying. So yeah, there is a difference in quality. I mean, otherwise you just have to clean them. Yeah? Both is void, yeah, just like in the stars. Yeah. So I mean, the image circle gets smaller on higher magnification, and notable it's 4x. Okay, I think I know what you mean. Okay, let me explain this as well. So let's go back here. So that's I'm opening this. Have a look what this is now. Basically, have a look what's going on here and also on the side. What I have is I've got so-called curl illumination. So what I'm able to do is I'm able to change the opening here of the this is called the field diaphragm over the lamp. Let's turn it down a little bit. Okay, so that's basically one thing. And if I open it all the way, I still get a circle. And what I can do in this case is I can have a so-called a flip out condenser. Look, I can flip it away. and then the circle disappears. The rainbow colors is because of the prism that I have, so I just remove those as well. Yeah. So what I can do, I can what I can do is I can actually flip in. Yeah. I can actually flip in the condenser and I can flip it out again. Okay. And this basically makes it appear in a circle. Yeah. Or not. Yeah. So that is essentially, maybe this kind of explains a little bit your question. And then in addition to that, I have the so-called field diaphragm, right? And the condenser aperture diaphragm, this changes the angle. But you know what? I don't know if I'm able to do that. I need a piece of paper for this. Let me see. Do a little, I don't know if I'm able to show this to you. I need to lower the stage here. You cannot see it properly. But look at it's if you look at the top, if I open the diaphragm, then you're actually able to see that by changing the condenser diaphragm, you're able to change the angle of the light. Okay, you have to look at right at the top. Okay, on the paper. Yeah. Yeah. Yeah, but now the angle is small and now the angle becomes larger, right? And if you open and close the field diaphragm, then you're changing the diameter. Yeah, it's a little bit difficult to see, but this is kind of a little demonstration on how different diaphragms are able to affect the light differently. Yeah, so it was just a little bit. Yeah. Next question: is UPROL still available? Yes, it is. Unfortunately, however, sometimes difficult to get. And yes, it is sold because people who are doing insect microscopy, they prefer to use UPROL. And I found it as well. You have to contact chemical supplies companies and laboratory supplies companies. So it's not a question of whether it's available or not, but sometimes they might not send to private people or individuals. So that's something that you have to figure out. Do they actually send it to private individuals, and are you willing to pay for it? That's the other question, right? Because sometimes it's not so cheap. Yeah, is it available? Yes, it is, because I've seen it. Yeah. What mountain has a high refractive index and is available to purchase? I think. Which one? I mean, there are many mountains available, and I think even Canada balsam is available, but you have to talk to specific companies that actually provide them, right? So that is more the question. You cannot buy them simply over Amazon. That's a little bit the issue, right? These are two. So you have to contact chemical supplies companies, laboratory supplies companies, and then they will have that. I generally recommend, if you're in Europe, you might want to check out Brunel microscopes in the United Kingdom, because they have got a lot of stuff, a lot of stains, mounting media, and so on, and they might be able to help you out. Okay, it's Brunel microscopes, it's called, and they're in the UK. Yeah. Okay, you can also look at two overlapping double slits. Okay. Yeah. Technically, the refractive index needs two mediums to compare, correct? Well, no, because if a medium has a certain refractive index, then that is already the refractive index that the medium has. Okay? However, in this case, it's, to have an effect on the light, the refractive index of the medium should be different than the refractive index of the specimen. Right? For example, for phase contrast microscopy, that is relative, of course, but the actual refractive index itself is actually a value that's assigned to the substance. Yeah. So, Carolina Biological Supplies has different mounting media. Thank you. Okay. So, what time is it? Okay, I still have I'm still going to do a little bit of stuff here. So I talked about this, Let me do the following just for the fun of it. yeah, why not put some artificial meat under the microscope? Because it looks also kind of interesting. Because in the past, at least, I was able to find some, how do you say, microplastics in it. I don't know if this is not the correct one that I am going to get out here. Let me see. But I need to do the following. I need to then probably change the, how do you say this around? Just a second. I need something to get in here. Here we go. Okay, let's take a. I should clean this as well first because I do not want to cross-contaminate this. Okay, fine. You take a small sample of whatever you want to take. This one is now preserved in alcohol, by the way. Yeah, just for your info, right? Yeah. So I can put the rest in here. I close it because there's alcohol all over the place, and alcohol evaporates. And I add a little bit of water. Ah, by the way, I know I like to sidetrack. Look, I don't have running water, so I keep my water in those plastic bottles. Do you see the difference here that there is some algae growing on here, right? It's discolored. That's why I switched over to black plastic bottles because they remove the light. And if there is no light visible, if no light to go in, then algae can also not grow. Okay, so that's a little bit the thing that I want to kind of clarify. I need a container now for some water. Okay, and let's. And what I'm doing right now is you take just a couple of tweezers, or let's say some toothpicks, and you just completely pick it apart what you've got here. Okay. And then, as a last thing to show you, I just want to put this stuff from the coffee machine under the microscope as well. Okay, so I think maybe that's enough. I'm going to use this here. It's a little too thick. So what I'm going to do is I'm going to lift it off. I'm going to remove those large pieces here. So the microscope, the cover glass is able to lie flat. Okay, and let's have a look at this here. So where is the scope view? Yeah. Completely lost the focus, of course. Okay. So, and this is basically how this artificial meat looks like under the microscope. And the things that I always like to look out for, and I have found quite a few of these things, is very often you find those fibers in there, those long string-like structures. Yeah, occasionally. And so you see that sometimes, yeah, these string-like structures, these fibers that you see, because these are soybean products, right? Sometimes, are microplastics. You find those fibers in so many different places in nature as well. Yeah. So, but that's simply something that I've been also considering as a citizen science project. A little bit, it is kind of the hunt for microplastics using microscopes. Yeah. Yeah, not everything is necessarily synthetic. It could also be natural fibers, but in some of these, you can find quite a few of these string-like structures. Yeah. Maybe you, for scanning the whole slide, you probably want to use a lower magnification, like here, for example, again. Especially on the polarized light, you can see the colors. Yeah, here again, some fibers. Yeah, just saying that, you find them all over the place. If you take a water sample, some sediment, you're going to find these fibers. It's, And there is a staining possibility that where you can actually detect whether it's really synthetic or not. Not tried it yet, but there are actually indeed protocols to detect microplastics. Yeah. So, so that's those round dots that you see, those round circles, these are fat droplets. So that's quite easily So in this case, these are not air bubbles or anything, but those round globules are fat globules. Yeah. So so it's I just try to show you a couple of how do you say this? Yeah. Exploring possibilities. That's a little bit also one of the reasons of this live stream. It's not always that the things that you see are super overwhelmingly cool or exciting or so. You see some more fibers. Yeah, this could be maybe on the surface of the cover glass. Yeah, but it simply should motivate you a little bit simply to start exploring the environment and learning how different things look like, even if if there's sometimes not always something super exciting to be found. Again, see over here a fiber. Yeah. So I've checked some time ago these artificial meats and also water samples and all sorts of things, and they kind of appear all over the place. It's kind of sad. Yeah. So just saying. So last thing that I want to do is I'm going to read a little bit, and then I want to scratch some of the stuff off from my coffee machine. Okay. Where can you get Nile red? That's a good question. Again, Nile red is the stain used to detect plastics. Okay. I did not use it yet, and I'd probably also have to. You know what? Okay. As a matter of fact, I do have a. I think I'm gonna talk to the chemistry department of my school. Okay, because they're getting all sorts of different chemicals for laboratory, and maybe they can actually tell me a little bit more about that. It's actually like this, that sometimes it's not even so easy to order certain chemicals. I will tell you the following. I once actually wanted to order something. It was I don't remember what, it was a completely harmless substance. And I wanted to order that, and I wanted to order it over the school that I work for, because we do have a chemistry department and so on. And what they wanted is they only wanted to send it away if we filled out a form, who is a safety form, who is in charge for the safety of the chemistry department. Only under these conditions were they willing to send us the stuff. And it was a completely harmless substance, right? So that is a little bit the problem that there is, that some of these companies only want to send to institutes, right? Institutions, and not to private individuals. Yeah. So that is the thing. Yeah. So a couple of questions here concerning what about specimens that has a lower refractive index than the medium? What would it look like if there is a difference in refractive index between the surrounding medium and the specimen? What you get is let's use this here as an example. You get patterns like this. So for example, you see those oil globules here, yeah. And also in the case, these are oil globules, but you can also see this a similar thing with air bubbles. If it's out of focus, you're gonna see a black ring around it, right? Yeah. So where is the arrow? Yeah. Do you see that those globules, if they're out of focus, have this black, and that's because the light is refracted. Yeah. And that's an artifact. So because this black ring really doesn't exist, right? So this means that if you have a difference in refractive index, you will see those patterns, and this can actually mean that the object sometimes is visible at all, right? Because it makes these dark shadows, right? Yeah. So, and here it's not quite as visible, but and if you open and close the condenser, then this will also have an effect, For example, now when you open and close the condenser, yeah, then it changes the light more than it does, yeah, because it also has basically then impact on the depth of field, right? And then sometimes these refractive patterns become more or less visible as well. So sometimes the difference in refractive index can actually cause these shadows around it, which actually make the objects better visible. Yeah. How does it taste? Oh, well, actually, it tastes a little bit like the, how do you say, it takes on the taste of the surrounding food, I would say. Yeah. Yeah. Why did you soak it in alcohol? That's a very easy, an important question to answer. Yeah, I soaked it in alcohol, otherwise I would not be able to keep it. If I were to soak it in water, then bacteria are going to start growing in there, and it's going to start decomposing and rotting and smelling bad, and it's gone, right? But if you soak it in alcohol, then water is going to be taken out, right? And bacteria are not able to grow, and then you can keep it practically indefinitely almost, right? Every now and then you probably want to exchange the alcohol so that any remaining moisture is also removed. Yeah, but that's for long-term preservation is always keep it in alcohol. Yeah. Okay. Yeah, similarity between optics in microscopy and astronomy. Okay, the Nile red is an issue, of course. I also had some problems getting it. Yeah. Opinion, we're all in different stages of our microscopy roadie, from total beginner to experts, even in adjacent fields with patience and respect. These streams have the beauty of unifying and motivating. Yeah, that's actually, I think one of the things is I think other people are also having some fun. I'm having a lot of fun in any case. Yeah. Did you hear about cleaning slides with a soap with no water at all? Hard soap, not liquid. I heard some people use this method, but you have to remove the soap somehow afterwards, right? So I not tried it. I can imagine that the hard soap, maybe because it's a little bit abrasive, maybe it takes away some of the things, but ultimately you have to take the soap away as well somehow. Yeah. Okay, we always use Alconox or Liquinox to clean glassware. Yeah. Here in Norway, I cannot order hydrogen peroxide with more than 3% without being put on a watch list. Yep. Yeah. Unfortunately, because hydrogen peroxide can be used to make explosives. And that's the reason why also in airplanes, you're not allowed to take along liquids, right? Yeah. Can depth of field be improved, or is that a physical parameter bound to a certain magnification? Its depth of field is not only magnification dependent, but also depends on the aperture. Right, so this is the way that you can improve depth of field is by doing focus stacking. Okay, so by combining different levels. However, having said that, I know that in many cases, who cares? Let's just use this here as an example, right? When I do the following, when I close the condenser aperture diaphragm, let's go up even further with the magnification, just to get my point across, right? When you actually close the condenser aperture diaphragm like this, the objects seem to be fairly crisp and in focus. And when I open it, it becomes brighter. And I turn down the magnification, it kind of looks a little bit washed out. However, it's important, and that's counterintuitive, the resolution sometimes is better. Okay? The contrast is lower, but you're able to see sometimes objects that are closer together. Okay, so you see there is this trade-off. And if you have close the condenser too much, like this, then sometimes you get refractive issues. And then you also get objects that are on top and on the bottom, both in focus. And then you cannot separate them anymore, yeah, vertically. Yeah. So while subjectively it might look better, sometimes objectively you're seeing less. I know this is kind of counterintuitive, right? Because more objects overlap and you have got stronger refractive patterns. Yeah. But it kind of looks better because the contrast is better. But actually you have to open it up more and go down with the light to actually see those structures that are in one level better, right? So actually you should be able to see more, right? But at a lower contrast. So you see that's a little bit counterintuitive, yeah? You could make videos on how microscopes work. I don't know much about the technical terms like depth of field, iris, etc. Thank you for the recommendation. Actually, this is actually a good point. However, if I do that, an introduction to depth of field, iris, Köhler illumination, diaphragm, field of view, okay? Field number of the eyepieces and so on. All of these terms, working distance, for example. Thank you for the recommendation. I will make a video. However, not in this channel, but in the other. I've got a sister channel called Microbehunter Microscopy, where I'm also doing microscopy review videos and so on. So I'm probably going to do the technical aspects there. Right? After that, you remove the soap with a dry cloth. Honestly, that's an interesting idea. I'd be worried a little bit about the soap being left on the slide because soap is a detergent and the pH generally is high. It's alkaline. I don't know if all of the water microorganisms actually like that. Just saying, okay? Depends quite a bit on what you're looking at for sure. But yeah, that's an interesting thing at least, but I have not tried it, so I don't want to comment on that. But I'm just having my concerns of soap being left on the microscope slide. Yeah? Diffraction occurs often depending on magnification. When stopping down too much, images will get softer and lose resolution. Yeah. So, yes, what time? Ooh, it's already 1 hour. But you know what? I want to know it now. Even, I don't know if I'm able to see something or not, but I want to know it. I want to put some of this stuff from inside my coffee thingy here. Look, this is let's go up here. This is this green stuff in here. I love it. I mean, that's one of the reasons why I don't like to clean those, how do you say, those containers. This is because I always consider it a little bit of a pity. Because I consider this so beautiful, those little things in here. So what I'm going to do is I'm just going to scratch some of this stuff here. I don't know how I'm supposed to get this out here. Let's scratch it. Ah, beautiful. Look, it's green. Okay, let's try this. Green is always good. I tell you why green is good, because green means chlorophyll, means photosynthesis, means possibly some algae. Okay. And I know what you're thinking. Some of you think, it's disgusting. Come on, man. You have no idea what we're eating. Okay, let's suspend it a little bit. It looks pretty good. It looks pretty good. I don't know. Let's have a look. I hope to find some diatoms. Honestly, I did not check it yet. Okay, so I really don't know. Okay, in here, and here we go. I always say where there is water, there is life. And scope view. Okay, let's see. It's a little too bright. Huh. Maybe are these diatoms? I don't know. It looks kind of very irregular. It's greenish. Some chlorophyll present. It's a biofilm in any case. I don't know. Let's go in a little more. Actually, I used to see something more in the coffee container of my mother's. We actually looked a little bit nicer. It depends always what is forming. Normally what I would do is I would just wait for a couple of minutes and see if oxygen bubbles at least something is moving here. Look, little tiny thing here. I know some of you are feeling disgusted right now. You do not want to know what you can find in your air conditioner, in the filter. And those tiny things that you see in the background moving around, many of these are bacteria. Calcium deposits definitely possible. That's definitely clear. And however, I'm quite sure that these are not only calcium deposits, because it's simply also there is a lot of discoloration going on here. And what I'm going to do now is so calcium deposits certainly increase the surface area for that. It's no question, right? And they add. Yeah, and here in phase contrast. Okay, Maybe those bright spots that you see could be maybe, I'm guessing, calcium crystals, but those darker ones in the background, many of them are bacteria. Yeah. Just have the right size and appearance. Yeah. I mean, I did find, some time ago in a coffee filter, really a nice collection of diatoms as well. I guess this depends also a little bit on the amount of light that's available. But I think we all agree there is a full ecosystem of stuff going on here. Cyanobacteria, maybe. Honestly, if you keep on looking hard enough, you're probably gonna find pretty much everything. Yeah, there's stuff there, but I have to also admit there have been more interesting. I've seen already more interesting things isolated from pots and pans and glass and glassware and so on. Yeah. Normally, what I would try now is I would just wait a little bit, a couple of minutes, to see if some oxygen bubbles form, because this is then an indication that actually photosynthesis is happening, and this also happens. Yeah. Yeah. But honestly, yeah, you get the idea. So I'm gonna. What time is it? It's 1 hour 36. I'm going to have a look at a couple of questions here. Okay. Yeah, sees a moldy tomato. Normal person, ooh, Microbehunter, yes. Yeah, so yeah, so that's definitely something. Feel disgusted about the right things. That's what I'm trying to say here. I mean, there is, to a certain extent, these are a natural part of our environment. Yeah. If a nematode worm is not frantically moving, does it mean it's dead? Not necessarily. It depends on the nematode worm. Okay, Paul is asking this already some time back. I mean, generally, movement is, of course, a sign of life, but I would not just conclude that just because it's not moving, it's automatically dead, right? Yeah. So this is something that but indeed, most or many nematode worms, they do move frantically, and if they're not moving, then could be that indeed there's a problem with those. Yeah. Okay. Yeah. I think hopefully I answered pretty much everything. I really want to see Hydro in the live stream. Yes, if I find one. Okay. Do you do fluorescent microscopy? No, I do not have fluorescent equipment. However, it is possible to do so-called blue light excitation microscopy, which basically means that if you shine blue light on certain algae, then they will start to glow red. However, this it's fluorescent microscopy. However, it's so faint that you need to take long-term exposure. Right, so it's not quite always suitable for a live stream, but I have experimented with that as well. So basically, you do not use UV light for fluorescent microscopy, but you use basically blue light, and then some algae, the chlorophyll is also able to shine, but you need to do a more long time exposure. Yeah, there was a question from Paul algae fungus. I may have missed the answer. Okay, I've not seen. I've missed the question. Okay, some question about. I'm going up the comment now. Maybe I've missed the question as well. Okay. I'm just going up the. Thank you. Oh, another donation here from Arthur. Thank you very much. Okay. Can microbes think? Or how do they know when they need to eat or where to go? That's an interesting question. It's a philosophical question. Let's put it this way. This is a very common question. Also, students of mine ask me this question. How do microbes actually know, under quotation marks, what to do? And I'm giving you this example. Might sound crazy, but I just want to give you an analogy. When I drop, I don't know, an object, I don't know, this water bottle here, right? And I drop it and let go, it falls down. How does the bottle know that it has to fall down, right? And the answer is it doesn't know. it basically just the forces acting on it. And complex behavior or seemingly complex behavior by microbes, that they're kind of moving towards oxygen, they're moving towards light, on the very basic level can be explained using chemical reactions. Okay? Which are forces of nature, so to say, right? So the term knowledge, as we use it for human beings, does not really apply to microorganisms, obviously. So how do they still know what to do? They basically, these are just responses to the laws of nature, so to say, right? Chemical reactions. And on the higher level, it kind of appears to be intelligent. Yeah? But actually, there is no real conscious, how do you say, awareness of it, right? It's a little bit like asking, just let's use our human body as an example. How do you know that your heart has to beat? Yeah, your heart is beating. Yeah. How does it know that, right? I don't know. I'm not consciously controlling it, right? So you see, and maybe that serves a little bit like an analogy of how microorganisms, under quotation marks, know what to do. No, it just happens, so to say, right? And this just happens is also not by coincidence that it happens, but can be explained in the terms of physical and chemical reactions. Okay, yeah. So it's a very interesting question. So thank you for it. It's a common question, a very interesting one, but I have to admit, maybe not always easy to explain. Yeah. Do you use cold fiber lights? Is it worth investing in it, or does it not matter? Cold fiber lights, I am not using it, but I have used a homemade version of this. Cold fiber lights is basically very often used in stereo microscopy when you use epi-illumination from the top. You have a box that makes the light, LEDs or something, and then using glass fiber, the light is kind of directed on your specimen. I don't use it. Is it worth it? It depends what you want to do. I would not use it for myself because it would be actually an additional box that I have to put up somewhere, and I don't have the space. And it's currently working with the setup that I have. Okay, I've got a stereo microscope here. However, if I were to use, let's put it this way. Let's say I want to use, just look at this one here, epi-illumination from, it is possible to look with a low power objective, to look at a specimen from the top, if you shine light from the top, right? And what I'm doing is I'm holding a flashlight. But if you want to do this on a more frequent basis, then I can indeed imagine that those fiber lights are actually very useful. Yeah. So I would say there are many ways how you can experiment around with, and there is not always a very objective answer. Okay, so my suggestion is if you think that you have the space and you want to experiment with this, then I highly would recommend it, right? Because people actually do use it. Yeah. So for my purposes, I decided not to use it simply because of space reasons. Yeah, and because I've got other ways that actually also kind of work. But I can imagine that there are some, some, how do you say, some uses for this. So, ladies and gentlemen, what I'm going to do now is the following. I would like to say thank you, okay? To AMRSH. Thank you very much for the contribution. I would like to say thank you. I hope I'm not overlooking someone, to Micropolo for your contribution. I would like to say thank you to Arthur S. For their contribution. Yeah, so thank you very much. I know today was again a little bit different, a little bit more improvised than maybe in the previous ones. If you have any certain wishes for me to do certain videos or to look at certain things, please do feel free to post it. I'm going to call it a day for today, and I think I'm just going to say goodbye and happy microbe hunting, and see you next week again. Okay, all the best and bye bye.